Welcome back to the Classics of Immunology Journal Club. And before we get started, don't forget to like the video and click on the bell to get notified for more, subscribe to the channel, and also check out my website and my social media sites. On the website, you'll find all of the classic papers, reports in immunology. And so if you read all those, you'll know more than most other people in the field. So today, we are focused down on a paper that came out in 1998 by this fellow right behind me here, Bruce Beutler, and his multiple students and postdocs and technicians and administrator who worked on this project. It took a long time, and I'll get into that. The title of the paper is Defective Lipopolysaccharide Signaling in C3H, HEJ, and C57 black 10 SCR mice, mutations in the Tlr4 gene. So it came out in Science in December of 1998. Now, the background on this paper is that Bruce had shown 13 years earlier in 1985 that LPS caused the production of TNF and other pro-inflammatory cytokines when he was a postdoc in Tony Cerami's lab at the Rockefeller University in New York City. And what he showed in subsequent papers, he purified the active component. Tony's lab had isolated or identified as a molecule that appeared in the serum of mice that had chronic infections and so forth that they called cachectin. Bruce purified that because his expertise at that point in time was primarily protein biochemistry. And he sequenced that at the N-terminus. And it was just at the time that a group out of Genentech, one of the first, if not the first, biotech companies, had cloned the cDNA and sequenced tumor necrosis factor. And when Bruce found essentially cachectin and TNF were exactly the same thing. And he went on then in a series of papers that were really classic. And we reviewed at least one of his papers, but if not more than that, where he proved essentially that LPS, which was called an endotoxin, was not a toxin. LPS stimulated the production of an endotoxin or a toxin by the host. And so the whole idea that microbes caused the disease an infectious disease and so forth, by releasing toxic molecules that were toxic to the host. By these experiments that Bruce had done back in the 1980s, essentially showed that wasn't true. Rather, the host was making the toxins. And that's very, very important in today's world of infectious diseases. Now, the question was that his experiments and so forth and Tony's lab exposed was, okay, well, LPS causes primarily macrophages to release these pro-inflammatory cytokines, TNF, interleukin-1, interleukin-6 being the three major pro-inflammatory cytokines. How did it do that? How did LPS cause the cells to make these these inflammatory molecules? It turns out that the paper we're reviewing today from 1998, which was 13 years after the landmark papers that Bruce published back in the 80s, 13 years after this 1998 paper, Bruce won the Nobel Prize, together with a couple of other people that I won't go into today, for his discovery of the receptor for lipopolysaccharide, or LPS. So the whole thing took 26 years from 1985 onward, and then there's a reason for that. In the introduction to this paper, Bruce goes into the history of this whole field, which goes way back, essentially, into the 19th century, and to the first awareness that microbes could cause disease, from Louis Pasteur and Robert Koch's laboratories in Europe. In the US today, and he points this out, as many as 20,000 people die every year because of infection with colonic organisms that are stain in a gram stain and negatively, so that they appear red in the gram stain. And it had been shown then that these organisms can cause septic shock, sepsis. Sepsis, if you look that up in the dictionary, it means rotten. It comes from Latin, meaning rotten. And sepsis is essentially what happens when you have bacteremia, when you have organisms that even starts out locally, but then gain entrance to the blood, and they multiply and multiply and multiply, ultimately causing septic shock, so that the blood pressure falls, and the mortality rate with bacteremia, with coloniformic organisms, is around 50%. Now, what had been shown between 1985 and 1998 in this paper is that as I pointed out, it wasn't the LPS that caused the shock. It was the TNF and IL-1 and IL-6 and all of those cytokines, because what they did was that they caused the capillaries to leak fluid from the blood system into the tissues. And if that became a systemic situation, so that it happened all over the body, then the blood volume was depleted very rapidly. And when you didn't have enough blood volume, you'd go to the brain and so forth, you went into shock, and then you died. Now, the question that was unanswered at the time of this 1998 paper was, well, is this septic shock situation, is that detrimental or is that beneficial to the host? And of course, now we know that if it's a localized infection and it's a localized inflammatory reaction, so that you have an infection underneath the skin in your arm, so to speak, for example, it gets red hot, swollen, and sore. Those are the cardinal signs of inflammation. If your whole body gets red, hot, swollen, and sore, ultimately you go into shock and you die. 30 years before this 98 paper, in 1968, as Bruce points out, there was a strain of mouse that was discovered. It was not susceptible to LPS-induced shock. You can give it LPS and nothing happened. And there was also a control mouse that was another strain. So that was called the C3H-HEJ mouse, and that was the one that was not susceptible to septic shock as a consequence of LPS injection. And its mate was C3H-HEN, which was susceptible. So that was very important because of the fact that meant that there might be a gene product, a gene that specified a gene product that was extremely important for the LPS effect. And ultimately that became called the LPS locus. About almost 10 years later, in 1977, another strain of mouse called the C57 Black 10 SCCR strain was also found to be not susceptible to LPS-induced shock. So that gave two different, and the genetics of the inheritance of this particular non-susceptibility to LPS was different from the C3H-HEJ strain. So that gave Bruce and his team the wherewithal to start to take a genetic approach to solving this situation as to what was the LPS locus doing. Now, in 1989-90, about another 10 years later or more, there was a cell surface protein that had a CD designation. CD14 was found to bind LPS. And everybody said, well, this is the LPS receptor. But the problem was there was no cytoplasmic domain to this protein. So how could this be the whole story in terms of it might bind it on the outside of a cell, but if it can't transduce a signal from the outside to the inside, then it really can't cause the cells to make this TNF and so forth. And so they figured then there must be another protein, an associative protein that did confer the signaling capacity onto the whole system. And that's what they set out to look for. Now, the LPS locus was mapped to mouse chromosome 4 between two different loci that were very far apart. And the problem with that that arose for Bruce and his team is that they had to really make up many of the methods of the genetic methods to try to find out what portion of what part of the whole genome was coding for the LPS, coded in the LPS locus. And what they did was they did shotgun clone genomic DNA, and they did it over 1.6 million base pairs. And over that total span, they really found over many sequencing experiments and so forth, which took a total, they set out their sights in 1993, and they didn't hit pay dirt until 1998. 5 years of no papers from the lab, and because the whole lab was focused on this particular problem. In the process of doing all the experiments they needed to do, they found only 2 genes in this total millions of base pairs that could code for real genes. There were a lot of pseudogenes, and there were a lot of blind alleys that they went down thinking that they might have it. But only 2 genes cropped up, and only one of them really panned out from the standpoint of the whole biology. And that was a gene that was coded for a protein in the toll-like receptor family that had been already identified by the geneticists in the fruit fly, Drosophila. The thing that made them think that maybe they'd finally hit it, was that the interleukin-1 receptor, which is a pro-inflammatory, interleukin-1 is a pro-inflammatory cytokine, that also was in the toll-like receptor family. So they honed in on the C3H-HEJ mouse and the C3H-HEN mouse, and they cloned cDNAs from these 2 different strains. And they then sequenced through over and over and over again, looking for a mutation in the HEJ strain that was not present in the HEN strain. And that gets us to the screen sharing. And here in figure 2, you can see what happened is that in the coding region, they found at position number 712, here's the C3H-HEJ, it codes for histidine, whereas HEN, the susceptible strain to LPS, it was a proline. If you look at the rat, it was a proline. If you looked at the human, it was a proline. And so they said, well, they've got it. This is it, hopefully. Because proline is a very important amino acid in protein structure. If you take that away or you change it, you can really change the whole configuration, the three-dimensional structure of the protein. They went on and they looked at the other strain, C57 Black 10 SCR, and this is a PCR gel looking for what they now knew was a 2.6 kb. They found it in the SEN strain, but it was absent from the CR strain. And when they looked at it northern blots, the CN strain, it was positive and it was clearly negative. So this mouse had a gene deletion, not just one amino acid change, had a gene deletion. And that really clinched it for them among a lot of other things that they talk about in the paper. So if we go on to the discussion and also what happened subsequently, in the discussion, they pointed out that in Drosophila, the toll signaling activates a gene that's called drosomycin, which protects the fruit fly from fungal infection. So they were in the same bailiwick, essentially. If you really want to understand exactly what happened, not only, well, in Bruce's life, as a young investigator growing up, trying to make his way and so forth, After he won the Nobel Prize in 2011, which wasn't too long ago, only 11 years ago now, and there's a wonderful accounting on the Nobel website, nobelprize.org. Look for medicine and physiology prizes and scroll down and look for 2011. You'll find Bruce's there. Click on his name. You'll ultimately get to two pieces on that website. One is the biography that Bruce wrote, and it's just really revealing. So if you're a young investigator thinking you might like to win the Nobel Prize someday, check out this biography. And then also look at the Nobel Prize winning lecture that follows right after the biography. This is when Bruce describes, after months and years of negative data, and a lot of people telling him that he was crazy to pursue this. And actually, he was chosen to be a Howard Hughes investigator, which, as you know, comes with a tranche of money that, together with NIH money, can really allow you to have a really good financial investment in your experiments. In April of 1998, now remember this was published in December of 1998. Howard Hughes people told him that they were cutting off his funding in 2 years, so it was going to expire in 2000. And so, such as life goes. It wasn't until September 5th, 1998, when Bruce was looking through the data from the day, the week's experiments, that they hit up. And he didn't know for sure, absolute sure exactly, but he thought this is it, based on the data that are there. And in the description in his biography, and also in the Nobel lecture on the science, you'll see he describes exactly what was going on. So that was the November 5th or September 5th. And they wrote the paper and well, they did some more experiments. Then they wrote the paper and they sent it off to Science on September 30th, 1998. And it was accepted right away and published in December. The reason that this finding and the whole history of this business is so important is that this really connected the mechanism of how the host senses the presence of foreign bugs, microbes. And it's through the sensation, the ability to sense molecules that were specific for the microbe. And not only to recognize or to sense that they're there, but to react against them. That's two different aspects of the whole thing and the signaling apparatus of that whole process. Because the way the host responded to this whole thing was that it created the immune inflammatory response. And that immune inflammatory response in the whole host, both mice as well as men, can lead to death. And so this underscores what I've been saying all along in the whole COVID pandemic, and that is that the symptoms that the people that brings the people to the physician, to the hospital, and to the intensive care unit, and then the morgue are not caused by the virus. It's not caused by COVID. All of the symptoms are caused by the host reaction to the presence of the virus. The reaction essentially is all mediated, all produced and mediated, orchestrated by the cytokine molecules, because then they bind to their receptors and so forth. We've been discussing for quite some time in this journal club, which also the reason that that's important is because that the therapy, if you have therapy like we do now with Paxlovid, for example, that will block the replication of the microbe, But if you don't have that, and we didn't have that at the beginning of the pandemic, what you do is that you try to block the inflammatory response. And you say, well, how do you do that? Well, the prototypic agent, and probably the first, was noticed in the 50s, were glucocorticoid hormones, which block, now we know, the ability of NF-kappa B to activate the pro-inflammatory cytokines. Now, the thing from the therapeutic standpoint is that if you've already got an inflammatory reaction that is ongoing and progressive, you can stop new production of TNF and IL-1 and IL-6. But we also nowadays have monoclonal antibodies that either block the cytokine itself or the cytokine receptor to take care of the cytokines that have already been produced. And that's what really should be doing. And I think it is finally, after 2 years, is part of the therapeutic armamentarium. Subsequently, what happened in this whole field is that, well, as soon as everybody was convinced that the LPS locus was coding for Tlr4, which was the molecule in question here, it was found that the mouse had genes for 12 TLRs. Humans had genes for 10 TLRs. They all bind and recognize different, multiple repeating patterns in microbial products. And so the whole idea of pathogen-associated molecular patterns, PAMPs, was introduced. And it was found that the TLR family of molecules were then subsequently called pattern recognition receptors, or PRRs. That essentially has nailed the fact that it's not the bug that's causing these symptoms. It's the cytokines. It's the reaction to the bug and the cytokines themselves that are causing those symptoms. So I'll stop here. And don't forget to like the video or the journal club and click on the bell and check out the website. And thanks for listening, and come back next week. If you're new to immunology, or you just want to delve into a certain aspect of this large field, and you want to get a grasp on things, you should buy my new book, Molecular Immunity, because it's a chronology of all of the important experiments and discoveries that have been made in the last 60 years. It's concise, but it's also very comprehensive. And it's the only such book that's been written since 1970. So you can buy it on Amazon, and together with the YouTube videos of the Classics in Immunology Journal Club, you can't go wrong.